Today I am going to discuss about chemical messengers. Chemical messengers, there are different types of chemical messengers. Some are locally active chemical messengers. Certain ones are neurotransmitters and neuropeptides, hormones and pheromones. All these chemicals act as messengers in human body. The local messengers, chemical messengers secreted by certain cells, and their action is on the cells around the cells which secrete these cells. This alters the physiological conditions in the immediate vicinity, acting on the nearby or neighboring cells. Act on the same cell, so the chemical messengers, the local messengers might be acting on the cell which secreted it. So then it will be called autocrine control, and it might be acting on the cells adjacent to the cells which secrete this one. So that sort of action is called paracrine action. And these local messengers do not accumulate in blood. So they will be synthesized as and when they are required, and they act on the same cell or the adjacent cells, and later they will be degraded. Example, lumones produced in the gut and regulate digestion. Histamine secreted by mast cells in wounds, and inflammatory response is produced in the wound region. Autocrine and paracrine control. Cells are secreting certain cells. There may be individual cells in isolated regions in the body, or there might be cells in different secretory glands. So they secrete the chemical messengers, and those messengers are released into the interstitial space, the space between the cells. So these messengers can either go and bind to the receptors on the same cell, so that is autocrine control, and cause the effect. Or these chemical messengers might be traveling through the interstitial fluid and reaching the receptors on the neighboring cells, bind to the receptors, and the effect will be produced, that is paracrine control. Hormones, they are signaling molecules synthesized within the body that regulate and coordinate physiological and metabolic functions. They act on receptors located on or inside the target cells. Sometimes the receptors are on the surface of the target cell, sometimes or the receptors will be located inside the cell. Produced by specialized secretory cells. So the hormones are produced by specialized secretory cells that are either localized in secretory glands. So they might be grouped in secretory glands like a thyroid gland, adrenal gland, etc., or within organs that have other primary functions. So like in pancreas, some hormone secreting cells are there. Pancreas also has some other cells. So depending upon the origin, that is from where or what type of cells are synthesizing these chemical messengers, and how they are transported. Are they transported through blood, or they are just released in the interstitial space, or whether they are transported through the neurons, axons of the neurons, depending upon these factors. So origin of synthesis, location of the synthesis, and the mode of transport. These hormones are given different names. The original definition of hormone was restricted to chemical messengers which are produced within glands of internal secretion. So originally hormones were classified as chemical messengers which are secreted in specific glands and released into circulation in small quantities to act on distant receptors. So their transport is through circulation only. That was the original definition of hormones. So, and this term hormone is derived from the Greek word hormene, to excite. So that was the function of the hormones, to excite physiological and or metabolic functions. Now, the definition of hormones are changed. Recent definition of hormones, they also include, in addition to the ones which are already included by the old definition, the new definition also include chemical messengers produced by other than specialized secretory cells. So they need not be produced by secretory cells. Any type of chemicals that can act as a messenger is named as hormones. And also signaling molecules that reach their target receptors by routes other than circulation. So the hormones can reach their target cells not only through blood but also through interstitial fluid or through neurons. So, as per the new definition, the hormones can act on receptors on the cell that produce them. And this type of action is called autocrine, which originated from the Greek word autos, meaning self. Then paracrine controls is already mentioned. Chemical messengers acting on the receptors of adjacent cells are called paracrine secretion. Greek words, the para means adjacent. Example, somatostatin in the pancreatic islets. So somatostatin is produced by certain secretory cells in the pancreatic islets. It acts on adjacent insulin and glucagon cells. So that is a paracrine control. This table shows certain chemical messages and their sources. So adrenal gland cortex secretes aldosterone, androstenedione, cortisol, dehydroepiandrosterone, DHEA, and estrone. Whereas adrenal gland medulla secretes enkephalins, epinephrine, norepinephrine, adrenaline, and noradrenaline. Gonads, there are two types of gonads, ovary and testis, and ovary secretes activin, androstenedione, inhibin, estradiol, estriol, estrone, FSH releasing peptide, progesterone, relaxin, etc. Whereas testis secretes activin, androstenedione, estradiol, inhibin, Mullerian inhibiting substance, testosterone, etc. Like that, different glands or secretory cells, different chemical messengers or hormones. Non-glandular sources of chemical messages. These include bone, brain, gastrointestinal tract, artery of heart, kidney, vascular endothelium, thymus, macrophages, monocytes, lymphocytes, placenta, skin, liver, muscles, platelets, etc. All these organs has got cells which produce chemical messages. For example, bone secrete bone-derived growth factor and osteocalcin. Brain secretes a number of chemical messengers like beta endorphins, corticotrophin releasing hormone, dynorphin, gastrin, growth hormone releasing hormone, insulin, leuenkephalin, luteinizing hormone releasing hormone, LRH, metenkephalin, oxytocin, somatostatin, thyrotropin releasing hormone, etc. So, like that, a number of chemical messengers are secreted by these non-glandular sources. And their mode of action, we have already discussed this. They can be autocrine, paracrine, and also endocrine action. In endocrine action, the chemical messengers are released into the circulatory system. Through blood, it reaches the target cell and act on its receptors. So, chemical messengers are transmitted to specific receptors through the extracellular fluid, across the synaptic gap, and through blood. When the chemical messengers are acting on the cells, synthesize them. It is called autocrine. Messages delivered to adjacent cells is paracrine, and circulated through plasma, it is endocrine action. Nerve cells deliver chemical messages also, in addition to their role in signal transduction, impulse transduction. So these delivery of chemical messages by the nerve cells are in two ways. In a paracrine fashion across a synaptic gap. So in a synaptic gap, so this cell, this neuron, the axon, this is an axon of one neuron. That neuron secreted this chemical messenger, that is a neurotransmitters. So that neurotransmitter is released at the synapse. So through the synapse, it reaches the receptors on the next neuron. So it is acting on the nearby cell, that is a paracrine action. And also it can be released into the blood. Through the blood it reaches different targets. So that is neuroendocrine action. So this is neurotransmission, one neuron to the next neuron, released by the presynaptic nerve of one neuron. Through the synaptic cleft, it reaches the postsynaptic dendron and binds to the receptors on the postsynaptic dendron. So that is neurotransmission. Here this is neuroendocrine action, released into the blood Through the blood, it reaches the target organ and causes the effect. So, neuroendocrine system. Endocrine system shares its signaling and coordinating function with the nervous system. So both these systems, nervous system as well as the endocrine system, perform the function of coordination of body's physiological functions. The two systems have evolved to control and integrate vital body function. That was their role. They are evolved to perform this function. That is the control and integrate vital body functions. Chemical messengers produced by nerve cells and released on the axonal endings usually act in endocrine or paracrine fashion. Neural signaling molecules released in the synaptic gap to activate receptors on the adjacent cell membranes are called neurotransmitters. Here, the chemical messenger secreted by this cell is released into the synaptic cleft. From there, this chemical messenger go and bind with the receptors on the postsynaptic dendron. So this is signaling molecules released into the synaptic gap to activate receptors on the adjacent cell membrane are called neurotransmitters. So these are neurotransmitters. You know that acetylcholine is a neurotransmitter. And specificity of the neurotransmitter. How it is decided. The specificity of the message is assured both by the specificity of receptors on the postsynaptic membrane. So this neurotransmitter is released here and it should go and bind with the receptors on the postsynaptic neuron or dendron. So there should be specific receptors on the postsynaptic dendron. So that will decide the specificity of neurotransmitter. So this can be different types of receptors on different postsynaptic dendrons. So depending upon that, the specificity is decided. If the receptor on the postsynaptic dendron is not specific to the chemical messenger released, no action will be produced. The messenger released will go without any use. And also the discrete physical alignment between the axon terminals of a specific type of neuron and the receptors on the postsynaptic membrane of a particular cell. So that physical alignment is also important. So the presynaptic neuron should come in contact with the postsynaptic neuron which has got specific receptors. Otherwise it cannot act. The neurotransmitters cannot act. The transmission of messages by neurotransmission is very fast. As in milliseconds, it can reach from one part of the body to the other end of the body. And it has got restricted points of delivery. So it will be delivered at the end of the axon. So it is a restricted point of delivery. Hormones act over a long period of time. So whereas neurotransmitter, it is released and the action is for milliseconds, and after that hormone transmitter is removed or destroyed. Whereas hormones, they have a longer period of time of action compared to the neurotransmitters. They act for seconds to hours compared to the milliseconds action of neurotransmitters, and are distributed diffusely through the extracellular fluid or blood to a large number of targets. So if they are released into the extracellular fluid, it will reach all the cells around. Or if it is going through the blood, it will reach throughout the body in a diffuse fashion because it gets diluted in the blood or in the intercellular fluid. So that is how hormones and neurotransmitters act. Neurotransmitters are fast and has got discrete targets, whereas hormones action is slower. They can act for a longer period of time and they get distributed diffusely to different targets. Neurotransmitter acetylcholine, you are familiar with this, released from the axonal endings of the motor nerve into the synaptic gap, and this will act on nicotinic cholinergic receptors on skeletal and cardiac muscle fibers. So when acetylcholine is released from the axonal endings of the motor nerve So it will be acting on nicotinic cholinergic receptors of skeletal and cardiac muscle fibers. Then this acetylcholine is released from parasympathetic nerves. This will act on muscarinic receptors in ganglia and glands. So if it is released from a motor neuron, it will acting on the nicotinic cholinergic receptors on skeletal and cardiac muscle fibers. If it is released from parasympathetic nerves, it acts on muscarinic receptors in ganglia and glands. So that is why that is what we have said in the previous slide. The physical contact between the neurons, pre and post synaptic neurons, is important. Sympathetic nerves release norepinephrine that act on catecholaminergic postsynaptic receptors. So this is important. Different chemical messengers, neurotransmitters, are binding to different types of receptors. So that is where the specificity comes. And also same type of neurotransmitter released by different types of nerves also will be binding on different types of receptors. That is due to the type of receptor present on neurohormones. These are also neural signaling molecules released into the circulation. They are called neuro endocrine secretions or neurohormones. So produced by the neurons but released into circulation. These messengers behave as hormones by their method of signal transmission. They will circulate through the blood and reach the target cell and act on the cells. So target cell can be at different part of the body. They reach the targets through the circulation. The specificity of receptors on target cell assure the delivery of endocrine messages to specific targets. As we have seen in the previous slide, the specific receptors need to be present on the target cell. Then only the neurotransmitter can, or any chemical messenger can, bind to the receptor and cause the effect. Example of neurohormones is antidiuretic hormone and oxytocin synthesized by the nerve cells residing in hypothalamus. And they are released into circulation from the nerve endings in the posterior pituitary gland, and they get circulated through the blood and produce their action. Whether it is neurotransmission or neurosecretion. Some cells disseminate chemical messages by a combination of endocrine and neural mode of action. So they will be disseminating chemical messages, and their action could be either endocrine or neural mode of communication. And you know that endocrine, what happens is released into the interstitial space or into the blood and get diffusely distributed along the body, and its action may last for minutes or hours. Whereas neural communication releases the axonal endings, and it will go to the postsynaptic membrane and bind to the receptors and produce action. So discrete action, discrete space, and for a short period of time. Example is the synaptic nerves communicate largely by neurotransmission of norepinephrine. So norepinephrine can act as a neurotransmitter, or some norepinephrine is released in the general circulation, act as neuroendocrine messenger. Then cells of adrenal medulla are developmentally and evolutionarily ganglionic neurons of the sympathetic nerves. So neurons which form the ganglion, and also is from the sympathetic nerves, and they lost their axons, and they release epinephrine and norepinephrine into the systemic circulation. This is an endocrine function of nerve cells. Their function altered from neurotransmission to neurosecretion because they lost their axons. So the neurotransmission is not possible. And now they are producing these chemical messengers which are released into the circulation. Example, epinephrine and norepinephrine. Hormones, eicosanoids like prostaglandins, thromboxanes, leukotrienes, and prostacyclins are group of signaling molecules included as hormones. So as per the new definition, these are also included in the group of hormones. They are short-lived chemical messengers. They exert autocrine, paracrine, and occasionally endocrine action on the receptors. So it can be on the same cell or neighboring cells or get distributed through the circulation or through the interstitial fluid and act on distant targets, acting as endocrine. So they exert autocrine, paracrine, and occasionally endocrine action on the receptors. That is, they act on the same cell which produce them or neighboring cell or get distributed to the different parts of the body and act on distant targets. That is endocrine action. The nitric oxide is the only inorganic signaling chemical messenger, and it controls a number of important functions in our body, including the antioxidant responses, inflammatory responses, etc. Then, structure and synthesis of hormones. By chemical structure, hormones fall into three groups: amines, peptides, proteins, and lipid derivatives. So, three groups: amines, peptides, or protein and lipid derivatives. Amine hormones are metabolites of amino acids. Example: epinephrine, norepinephrine, and dopamine. They are collectively called catecholamines, synthesized from the aromatic amino acid phenylalanine and tyrosine in the brain and in the adrenal medulla. Frequently, hormone synthesis is under the control of other hormones. Example: cortisol controls the last enzyme phenylethanolamine N-methyl transferase (PNMT) in the synthesis of epinephrine. So this is an example of one hormone controlling the synthesis of another hormone. Epinephrine is synthesized from the aromatic amino acid tyrosine and phenylalanine. And you know that phenylalanine is an essential amino acid that has to be obtained in the diet. Sympathetic nerves and central nervous system synthesize norepinephrine. Adrenal medullary cells produce norepinephrine and epinephrine. So these are some examples of hormone production and their sites. Norepinephrine can be converted into epinephrine in the medulla, and this is facilitated by the adrenal cortical hormone cortisol. So this is another example of a hormone controlling the synthesis of another hormone. Cortisol is controlling the synthesis of epinephrine. This is an example of synthesis of two hormones, synthesis of catecholamines. So it starts from phenylalanine in the presence of the enzyme phenylalanine hydroxylase. It will be converted into L-tyrosine. This tyrosine is converted into L-dopa with the help of the enzyme tyrosine hydroxylase. L-dopa is converted into dopamine. This is also acting as a chemical messenger. L-aromatic amino acid decarboxylase is needed for the conversion of L-dopa to dopamine. And this dopamine can be converted into norepinephrine by the catalytic action of dopamine beta hydroxylase. So norepinephrine is another chemical messenger which can also be converted into epinephrine by the catalytic action of phenylethanolamine N-methyl transferase. So dopamine, norepinephrine, and epinephrine are produced. Storage and release of hormones. There are two types of hormone release. So the one is shown in this picture. Here the hormone is produced, stored in secretory vesicles until and when it is needed and then released. Here hormone is produced and released immediately. So hormones are released mainly in two ways. Amine and peptide hormones are stored in storage vesicles before release. So they are synthesized and stored in storage vesicles. They will be released. So the control is over the release of the hormone. That is how the hormone action is controlled. This secretion is controlled by neurotransmitters, hormones, and metabolites. Neural influence imposes a rhythmicity to hormone secretion. So there is a rhythmicity, the period of which depends on the type of hormone and the type of action needed. That we will see later, details. Various secretagogues, many factors which control the secretion, promote fusion of vesicles and cell membrane. So the vesicles come to the cell membrane was and when they need to be released. So this is controlled by various secretagogues, various factors which controlling the secretion. So as a result, the vesicle will fuse with the cell membrane and they break. The wall of the vesicle rupture and exocytosis will happen and hormones will be released either to the interstitial space or into the circulation. Second type of control, steroid hormones leave the Golgi apparatus in transport vesicles. So here the hormones are synthesized when they are needed. So the control is in the synthesis process. In the previous one, the control was on the during the release process. The hormone is constitutively produced and stored in storage vesicles in the previous case. And they will be released when they are needed, controlled by the various secretagogues, various factors controlling this secretion. Here, the hormones are produced when they are needed. They will not be stored in the body. And they are released into circulation by exocytosis without being stored within the cytoplasm. So produced, transferred to transport vesicles. The vesicles straight away move to the cell surface, fuse with the cell membrane, and vesicle rupture and exocytosis happens. So here the control is in the synthesis. Control of hormonal response is at the stage of hormone synthesis rather than hormone release. So there are two types of storage and release of hormones. One, the hormone is synthesized and stored in vesicles and they will be released when they are needed. In the second, the hormone is produced at the time of necessity and immediately they will be transported to the cell membrane and released to the circulation or the interstitial fluid. Then regulation of hormone release. Peptide and amine messengers are stored in secretory vesicles. We see the first type of secretion. The release requires transduction of hormonal, neuronal and metabolic stimuli by way of intracellular calcium or second messenger. So by controlling the intracellular calcium level or by using a second messenger, the release of these hormones from the secretory vesicles are controlled. That is the release of peptide and amine hormones or messengers. The release of steroid hormones is constitutive. Here hormonal, neuronal and metabolic stimuli control steroid hormone synthesis. Immediately after synthesis, they will be stored in transport vesicles or they will be moved into transport vesicles or they will be filled in transport vesicles and vesicles straight away move to the cell surface membrane and rupture and release into the blood circulation or into the interstitial space. So immediately the hormones are released from the transport vesicles. So these are the two types of hormone release. Then how much is released? That is also important. They are released in nanomolar or picomolar concentrations, very low concentration. So the action and the specificity depends upon the specificity of the receptors to the hormones or their binding capacity. So these released hormones in picomolar or concentrations or nanomolar concentration circulated in the blood at such a very low concentration. Success of hormone mediated response transmission rely on the specificity and high affinity of their binding to the receptors. So the specificity of the receptor to the hormone or the messenger and also the high affinity. If the affinity is high, more action. If the high affinity is low, the action will be slower and lesser. Most hormones are secreted in intermittent rather than continuous fashion. So hormones, almost all hormones are released in an intermittent fashion, not continuously. It has a specific reason that we will discuss in coming slides. Most hormones are secreted in intermittent rather than continuous fashion. So they are not continuously released. So you know that it is released in picomolar or nanomolar concentration and continuous release will cause dilution of the hormones. But if they are released intermittently and enough quantity can be released that time and that serves to supply hormones in quantities required for their action. The frequency of basal hormone pulse ranges from minutes to hours. So it can be released in every minute or in every hour, once an hour, something like that. There are also seasonal rhythms of hormone release. For example, certain hormones are released during different stages of menstrual cycle. Various stimuli can acutely increase or decrease hormone secretion. Example, acute change in secretion of some hormones during exercise. So the stimuli, different stimuli are there. They can acutely increase or decrease hormone secretion. Then compensatory changes in hormone secretion usually result from metabolic, neuronal and hormonal feedback. So these feedbacks are quite important. That controls the compensatory release in the hormone secretion and release. Different stimuli such as metabolic, neural and hormonal feedback cause compensatory change in the secretion of the hormones. They can increase or decrease the secretion of hormones, these stimuli. This feedback control is over the normal hormone secretion signaling. So this compensatory changes is above the normal hormone secretion. The secretion rate is out of alignment with the acute need for hormone secretion. So when the secretion rate is not in agreement with the acute need of the hormone, this compensatory mechanism come into action. These different stimuli, metabolic stimuli, neural stimuli, hormonal stimuli cause excess secretion to meet the acute requirement, need of the hormone secretion. Pattern of hormone release and its effects. The pattern in which the hormone is released has an important effect on the target organs because of the nature of the receptors. The temporal pattern of hormone secretion is of remarkable biological significance, and we are going to see what it is. The same quantity of hormone can have opposite biological functions when it is delivered in intermittent as opposed to tonic fashion. So, one situation the hormones are released in intermittent fashion, other situation tonic. The same hormone is released continuously. So the effect produced by these two types of release is quite different. Sometimes tonic release will not make any difference. For example, when growth hormone is released continuously, it has no effect at all. Only we will think if growth hormone is available continuously, there will be more growth. But actually, when growth hormone is continuously released, that's tonic administration of growth hormone, has no effect on growth. Normal menstrual function is achieved by a circular release of LH. That is, release of LH every hour causes or results in normal menstrual function. If the LH release is lower or higher or continuous, it will suppress menstrual cycle. So the temporal release of or the pattern of release of hormone is quite important. Modulation of biological response by changes in the temporal pattern of hormone secretion is often exploited for specific purposes. So if you know the effects, how the release of hormone causes the change in effect, if you know it, you can use this for specific purposes, for even for treatment purposes. For example, tonic administration of gonadotropins has been successfully used to achieve contraception. How hormones are transported? They are transported in two ways. One, bound to certain materials, or they are transported freely. Peptide, amine and protein hormones are water soluble. So they are dissolved in water and they can easily be transported through the interstitial fluid or through the blood. So they are circulated freely. Whereas all of lipophilic steroid hormones, they are not water soluble. Then growth hormone and some growth factors circulate bound to carrier proteins. So they are bound to carrier proteins and they will be transported. Albumin and pre-albumin, they can non-selectively bind to and transport a variety of small messenger molecules. So many small messenger molecules are transported bound to albumin and pre-albumin. This binding is non-selective. Globulins have single high affinity binding sites for specific steroid and amine hormones. So different types of binding materials are available and the chemical messengers are bound to them and transported. So is there any difference in the action between the freely transported hormones and the bound hormones? We will see in the next slide. So what is the importance of hormone binding to carriers? So binding your hormones to carriers extend the period of their availability and action by one, preventing their rapid clearance from plasma. So they will not be cleared rapidly from the plasma. They will not be easily destroyed or excreted. And second is keeping some of the hormone from circulating in the free form. So only a small amount is released, and so they can be released for a longer period of time. Extend the duration of hormone action, but the action will be reduced, but reduced action for a longer period of time. That is another advantage of binding hormones to carriers. Then insulin-like growth factors are protected from degradation, and the duration of their biological action extended for hours by the complex system of binding protein. So this complex system of binding proteins bind with insulin-like growth factors and protect them from degradation. Otherwise, they will get degraded immediately or in a faster pace. So this degradation will be slowed or protected from degradation by binding with the complex protein, and their action will be available for a longer period of time. In some instances, for example, growth hormone, binding proteins have almost identical structure to cell receptor, and this facilitates binding of the hormone to the receptors. So the binding proteins have receptor sites. The binding proteins have identical structures to the cell receptors, and they facilitate the binding of the hormone to the receptor. Thus, the binding proteins may facilitate hormone action. Then, how are hormones degraded? Or is it necessary for hormone to be degraded? To be most effective, hormones need to reach their target receptors in intermittent fashion. Why? Receptors become down-regulated or less sensitive to hormone. So receptors can become down-regulated or become less sensitive to hormone action. When, in two situations, this can happen. Hormones are present in unphysiologically high concentrations. Then also for a longer, abnormally long period of time. These two situations, high concentrations as well as availability for a longer period of time, will down-regulate or make the receptor less sensitive to the hormone. So that is why the hormones are released in intermittent fashion. They are not continuously released, and also they are bound to, many hormones are bound to different binding agents. As a result, they will be released in a smaller concentration. So it will not reach unphysiologically high concentration. So that will protect the receptors. Hormone degradation also avoid this sort of situation, because if it is high concentration, the more will get degraded, and also after some time all the hormones will be degraded. So these two conditions are not reached normally in a physiologically functioning body. Hence it is necessary prerequisite. So it is a necessary prerequisite for optimal hormone sensitivity and action, because the hormone need to be released intermittently and in smaller quantities. And also they need to be degraded. Otherwise, what will happen? Even though the release is intermittent, after some time the concentration will keep on increasing. Hormones are changed into metabolically inactive form, either in target cell or in liver and kidney, whether the enzymes are present. As the enzymes for inactivation is present. Hormones are changed into metabolically inactive form. This change can happen in the target cell or in the liver and kidney. If it is circulated through blood, it will reach liver and kidney. There they will be made metabolically inactive, because the enzymes for this process making them inactive is present in liver and kidney or in the target cell. Wherever these enzymes are present, there they get inactivated. A number of enzymes participate in the metabolic degradation of hormones. The speed with which the hormones are metabolized varies. It depends on whether they travel free in plasma. If they are traveling free in plasma, the degradation will be faster. If they are bound to any binding agent, the degradation will be slower. The molecular structure of hormones also affects their degradation pattern. Certain structures are not prone to degradation, but some others are easily prone to degradation. The half-life of hormones vary from seconds to hours. Half-life is the time required to get the hormone degraded to half the quantity. So it varies from seconds to hours, and the next table will see some half-life of certain chemicals. Eicosanoids, the half-life is seconds to minutes. Amines, minutes. Thyroid hormone T4 has a half-life of 6.7 days, whereas the another thyroid hormone T3 has a half-life only 0.75 days, around 16 hours. Polypeptides, 44 to 40 minutes. Proteins, 15 to 170 minutes. Steroid hormones, 4 to 120 minutes. So proteins have 15 to 170 minutes of half-life because different proteins have different structures. As I said in the slide before, depending upon the structure of the hormone or chemical messenger, the degradation time, the speed of degradation also varies. So this is the short description of chemical messengers. So you need to answer the questions. Hormone needs to reach their target receptors in intermittent fashion. Why? The degradation of hormones are quite important for maintaining the receptor activity. Explain. What are the different modes of release of hormone? How are they controlled? What is the difference between neurotransmitters and hormones in their action? What are the different modes of release and transport of hormones?